Welcome to today's lecture on the fascinating topic of muscle regeneration. In this session, we will delve into the intricate processes and mechanisms that underlie the regeneration of skeletal muscle following injury or damage. Muscle regeneration is an intricate and complex phenomenon that involves the orchestrated interplay of various cell types, signaling molecules, and extracellular matrix components. Understanding the intricacies of muscle regeneration is essential for developing novel therapeutic strategies for muscle injuries and diseases. So, without further ado, let's dive into the world of muscle regeneration and explore its different aspects. Muscle repair and regeneration are facilitated by satellite cells, which are stem cells located beneath the basal lamina surrounding muscle fibers. Upon injury, satellite cells activate, proliferate, and differentiate into myoblasts, which fuse to reconstitute damaged muscle fibers. This process is regulated by various molecular cues and signaling pathways. Satellite cells also have self-renewal capacity and contribute to muscle growth. Ongoing research aims to uncover their regenerative mechanisms. Macrophages are immune cells that help with muscle regeneration. They clear debris, recruit other immune cells, and promote tissue repair. They also remove damaged cells and help with the formation of new blood vessels. Their interaction with other cells is important for muscle regeneration. During skeletal muscle regeneration, the extracellular matrix, ECM, undergoes dynamic changes to support tissue repair. Proteolytic enzymes degrade damaged matrix components, while fibroblasts secrete ECM proteins to create space for new tissue formation. Signaling molecules like TGF-beta and IGFs modulate this remodeling process, crucial for functional muscle regeneration. The process of myogenic differentiation, in which myoblasts give rise to mature muscle fibers, is tightly controlled by a myriad of molecular mechanisms. Key regulatory factors, such as MyoD, MIF5, PAX7, and MRF4, orchestrate the progression of myoblasts towards the myogenic lineage. These factors activate the expression of muscle-specific genes, such as those encoding contractile proteins, leading to the formation of functional muscle fibers. Numerous signaling pathways, including the Notch, WNT, and TGF-beta pathways, interact with the myogenic regulatory factors, modulating their activity and influencing myoblast fate determination. Additionally, epigenetic modifications, such as DNA methylation and histone modifications, play crucial roles in controlling gene expression during myogenic differentiation. A comprehensive understanding of the molecular mechanisms governing myogenic differentiation holds significant promise for therapeutic interventions targeting muscle regeneration and repairing muscle-related pathologies. In muscle regeneration, the formation of new blood vessels, called angiogenesis, is crucial for delivering oxygen and nutrients to the regenerating tissue. This process involves the upregulation of pro-angiogenic factors that stimulate the growth of new blood vessels from pre-existing ones. Endothelial cells, which make up blood vessels, undergo changes to create a network of capillaries in the regenerating muscle. The interaction between these cells is important for vascular stability and functionality. Disruptions to angiogenesis can impair muscle regeneration, highlighting the need to understand these mechanisms for developing better treatments. Growth factors play a significant role in promoting muscle regeneration by regulating various cellular processes, including cell proliferation, migration, and differentiation. Several growth factors, such as insulin-like growth factors, IGFs, hepatocyte growth factor, HGF, and fibroblast growth factors, FGFs, have been shown to stimulate satellite cell activation and enhance myoblast fusion, thus facilitating the regeneration of muscle tissue. Mechanistically, these growth factors activate intracellular signaling pathways, such as the PEAK-ACT-mTOR pathway, the MOP/ERK pathway, and the SMAD pathway, to mediate their effects on muscle cells. Additionally, growth factors can regulate ECM remodeling, angiogenesis, and immune cell recruitment, further influencing the regenerative process. Harnessing the potential of growth factors in promoting muscle regeneration holds promise for developing novel therapeutics and interventions for muscle-related disorders. Stem cell-based therapies offer promise for muscle regeneration. Embryonic stem cells and induced pluripotent stem cells can differentiate into muscle cells and contribute to regeneration. Adult stem cells promote myogenic differentiation, modulate inflammation, and support blood vessel growth. However, challenges remain for clinical translation. Innervation, or nerve fiber regrowth, is crucial for muscle regeneration. Regenerating muscle releases molecules that guide nerve fiber outgrowth. Motor neurons extend axons towards regenerating muscle, forming connections at neuromuscular junctions. Molecules like BDNF and GDNF aid in nerve fiber regeneration and NMJ formation. Understanding this interplay is important for functional recovery. In the video on muscle regeneration, we explore how epigenetic regulation affects the expression of genes. Epigenetic modifications, such as DNA methylation and histone modifications, control gene activation and repression during muscle regeneration. This complex process is influenced by transcriptional regulators and signaling pathways. Dysregulation of epigenetic processes can impair muscle regeneration and contribute to muscle-related disorders. Excitingly, understanding the epigenetic landscape opens up possibilities for targeted therapies to enhance muscle repair. Cytokines and chemokines are small molecules secreted by cells that regulate inflammation and tissue repair. Pro-inflammatory cytokines recruit immune cells, while anti-inflammatory cytokines resolve inflammation. Chemokines help immune cells and other cells migrate to damaged tissue. Understanding these processes could lead to new muscle repair treatments. As individuals age, the ability of skeletal muscle to regenerate declines. Factors such as a decrease in satellite cell functionality, altered extracellular matrix composition, reduced angiogenesis, impaired immune response, and changes in systemic factors like growth factor availability and chronic inflammation contribute to impaired muscle regeneration. Understanding these mechanisms is essential for developing effective interventions for promoting muscle repair in the elderly. Tissue engineering offers innovative approaches for muscle regeneration, using biomaterials, cells, and bioactive factors. This includes creating bioengineered scaffolds that mimic natural muscle tissue, delivering growth factors and cytokines, and using cell-based therapies. These strategies have the potential to revolutionize muscle regeneration. Muscle regeneration is a complex process involving various cells, molecules, and matrix components. Understanding it helps develop therapies for muscle injuries and diseases. Our goal is to improve muscle regeneration and functional recovery through satellite cell targeting, immune response modulation, growth factor manipulation, and tissue engineering.